Hello all. Today we are going to discuss about bacterial food infection. This bacterial food infection occurs when a bacteria contaminate food and cause illness in individual who consume it. There are different types of bacterial food infection and they are salmonellosis, campylobacteriosis, E. coli infection, listeriosis and more. Let's discuss each one by one. Before that we have to know about what is food infection. This food infection, which is also known as foodborne infection, which refers to a type of foodborne illness caused by the consumption of food contaminated with pathogenic microorganisms such as bacteria, viruses, or parasites. This infection occurs when a pathogen enters gastrointestinal tract and multiplies in number. These bacteria penetrate into intestinal mucosa and grow in number, which pass through systemic organs. The next one is zoonotic disease. What does the term zoonotic disease means? These are illness that can be transmitted mainly from animals to humans. These diseases can be caused by various pathogens including bacteria, viruses, parasites and fungi. According to World Health Organization defines zoonosis or zoonosis as those disease and infection which are naturally transmitted between vertebrate animals and man. And the mode of transmission of these zoonotic diseases mainly through feces, urine, saliva, blood, milk, oral contact with bedding or animals. Approximately about 150 zoonotic diseases are known to exist. Some examples of zoonotic diseases are rabies, Lyme disease, salmonellosis, avian influenza, leptospirosis, etc. Rabies can be transmitted directly from animals to humans through dogs. But some diseases can be acquired indirectly by human through ingestion, inhalation, or contact with infected animal products, soil, water, or other environmental surfaces that are contaminated with animal waste or a dead animal. Example, salmonellosis, leptospirosis, and anthrax. The infected chicken meat can cause Campylobacter infection. Some zoonotic agents can cause listeriosis and E. coli gastroenteritis, and few diseases that are transmitted with the help of vectors such as mosquito or other arthropod which can transmit disease to humans. Example, St. Louis encephalitis, then Rocky Mountain spotted fever. This St. Louis encephalitis is a kind of a viral disease that affects brain which is transmitted by mosquito. And the virus transmitted is flavivirus. Next is Rocky Mountain spotted fever. This is a bacterial infection caused by Rickettsia rickettsii, which is transmitted to human through bite of an infected tick. Now let's discuss each disease condition one by one. First one is salmonellosis. This is a bacterial infection caused by Salmonella bacteria that affect gastrointestinal tract. The causative organism which is important, Salmonella gastroenteritis. This is caused by the ingestion of food that contain particular number of viable cells, that is living cells of members of the genus Salmonella. Then what are the characteristics of Salmonella? This is a small gram-negative bacteria, motile, which that is they can move from one part to another. The non-spore forming rod that ferment glucose and produce gas. They do not ferment lactose or sucrose. The salmonellosis can be caused due to the consumption of 10 raised to 6 to 10 raised to 9 bacteria. The source of infection is the bacteria in intestinal tract of animals such as birds, reptiles, farm animals, humans, and occasionally insects. Also, there are some other mode of transmission like foodborne that are contaminated food including poultry, egg, meat, and dairy products. Waterborne that is contaminated water with the feces of animal. Then animal contact, that is direct contact with infected animals. Then person to person, it can transmit from infected person to a healthy person. Infected rodents, rats and mice may contaminate unprotected foods with their feces and thus spread salmonella bacteria. And flies can also play an important role in the spread of salmonella, that is through especially contaminated fecal matter to foods. As humans acquire this bacteria through contaminated foods such as beef, poultry, egg products, or water. The major symptoms of salmonellosis can occur longer incubation period. The salmonellosis is usually about 12 to 36 hours and staphylococcal poisoning is about 2 to 4 hours. Only after this incubation period they start to show these symptoms such as nausea, vomiting, abdominal pain, headache, then diarrhea, namely watery, greenish, foul smell stools, faintness, moderate fever, prostration, muscular weakness, then restlessness, twitching, and drowsiness. The mortality cases in salmonellosis is very few, that is less than 1%. Indisability in the sense gastrointestinal instability refers to a condition where the intestines are unable to function properly that can lead to several symptoms. In case of salmonella, it can cause slight discomfort and diarrhea to death in 2 to 6 days. About 0.2 to 5% of patients may become carrier of the salmonella organism. In case of acute phase, per gram of feces contain about 1 billion of salmonella bacteria. And please note down the associated foods, that is raw meats, poultry, egg, meat, then milk and dairy products, fish, shrimp, coconut sauces and salad dressings, cake mixes, cocoa, peanut butter, and chocolate. Then, how can we prevent the outbreak of salmonellosis? Heating the food is the best method so that all portions should reach about 66 degrees Celsius for 12 to 15 minutes. That can distract most of the salmonella resistant types. Then refrigerating all the food susceptible to salmonella contamination. Then avoid prolonged holding of this food at a room temperature that enhance the growth of salmonella bacteria. The next important bacterial infection is enteropathogenic Escherichia coli. This enteropathogenic E. coli, that is known by EPEC, is a type of Escherichia coli bacteria that can cause diarrhea and intestinal infection that mainly affect infants and young children. The human diarrheal disease or food poisoning outbreak can cause enteropathogenic E. coli that can grow at a temperature range of about 20 to 40 degrees Celsius with the minimum growth temperature at 10 degrees Celsius and optimum at 37 degrees Celsius. This heating at 65 degrees Celsius for 15 to 20 minutes can kill all the E. coli. That is meant by the term lethal. And the important one is pH range for the growth is 4.2 to 8.5 with the optimum in the range of pH 7.2 to 7.5. That is highest number of E. coli bacteria can grow in 7.2 to 7.5. Even E. coli can grow in the presence of 5% salt at 35 degrees Celsius and it can be inhibited by 10% of salt at 37 degrees Celsius. The symptoms of E. coli can be caused by the ingestion of 10 raised to 6 to 10 raised to 10 viable or living cells per gram that must colonize the small intestine and produce enterotoxin. The major symptoms include non-bloody watery diarrhea without inflammatory in stools. Incubation period is about 2 days after eating the contaminated food and it can last for 8 days. Also, it can cause cramps, chills, vomiting, aches, and headaches. Few other symptoms are there like a blood in stools, there is visible blood in stool or black terrace stool, then weight loss, dehydration, etc. The foods associated with E. coli infestation include cream, pie, mashed potatoes, cream puffs, and creamed fish. Then consumption of a milk, cheese, ice cream, meats, fish, and macaroni. And these E. coli can be destroyed by drying or freezing, but some of the spores can exist for a particular period. There are different types of strains of E. coli. First one is enteropathogenic strains of E. coli. As we have already discussed, it can affect intestine tract that they colonize in small intestine and cause acute gastroenteritis in newborn and infant up to the age of 2 years. This enteroinvasive strains of E. coli can affect epithelial cells of large intestine that in turn cause diarrhea in older children and adults. And the enterotoxigenic refers to type of microorganism that produce toxins which affect intestinal tract and cause diseases. There are two types of toxins are producing: heat stable toxin and heat liable toxin. This heat stable toxin can withstand high temperature, but in case of heat liable toxin, they denatures when high temperature is applied. Both these toxins cause diarrhea in adult and infant. This E. coli is also called as traveler's diarrhea, which is diagnosed by the isolation of bacteria from feces. The treatment for E. coli infestation can be applied by fluid and electrolytes. This enteropathogenic E. coli can be prevented by avoiding contaminated food and water that have high coliform count, avoid unpasteurized juices, wash fresh fruits and vegetables thoroughly before eating in raw form, then use adequate cooking procedure for the destruction and prompt refrigeration. This E. coli infection can withstand up to 5 to 10 days without treatment. Then the consumption of antibiotics and antidiarrheal drugs are not helpful to prevent E. coli infestation. Next bacterial infection is Bacillus cereus gastroenteritis. This Bacillus cereus is a type of a food poisoning, a type of gram-positive aerobic bacteria, spore-forming rod-shaped one, mainly present in soil, dust, and water. The minimum growth of the bacteria is about 4 to 5 degrees Celsius temperature with maximum of 48 to 50 degrees Celsius. The optimum pH range is about 4.9 to 9.3. The symptoms can occur due to the consumption of large number of viable cells such as 108 per gram of Bacillus cereus must be ingested to develop signs and symptoms. Mainly acute abdominal pain, flatulence, that's gas formation, and watery diarrhea. Headache and dizziness are common, then dehydration and prostration may occur, but nausea, vomiting, fever, and chills are very rare. The incubation period for Bacillus cereus infestation is 6 to 15 hours after consumption of food and not more than 24 hours. The foods that affect Bacillus cereus gastroenteritis are cereal dishes that contain corn and cornstarch, mashed potatoes, vegetables, minced meat, liver, sausage, milk, and cooked meat. And some food mixtures such as sauces, puddings, soups, pastries, and salads have frequently been incriminated in outbreak. So let's move on to cholera, which is a gram-negative bacteria. This is a kind of highly infectious diarrheal disease mainly caused by the bacterium Vibrio cholerae. By ingesting food or water contaminated by fecal material from patients or carriers. Also, this shellfish and planktons are the natural reservoir of Vibrio cholerae. Let's discuss about the symptoms of cholera. Once the bacteria enter the body, incubation period is from several hours to 3 or more days. An infected dose of around 1 million organisms should be ingested to cause illness. These bacteria enter into small intestine and are attached to the wall and can secrete cholera enterotoxin called that is choleragen. As a result, there is a hypersecretion of water and chloride ion while inhibiting absorption of sodium ions. Excretion of fluid and electrolyte with associated abdominal muscle cramps, vomiting, fever, and watery diarrhea, and the person can lose about 10 to 15 liters of fluid during infection. The higher concentration of a blood protein caused by reduced fluid level, which can lead to circulatory shock and collapse, then death. The incubation period may vary from 6 hours to 5 days. Associated foods include spread of poor sanitation results in contaminated water supplies. Then sporadic cases occurs when shellfish harvested from fecally polluted coastal waters which are consumed in raw form. It can be diagnosed by the isolation of causative organism from diarrheic stool of infected individuals. Prevention of cholera can be done by drinking boiled water or treated with chlorine or iodine water. Then other safe beverages include tea and coffee made with boiled water and carbonated bottled beverages with no ice. Eat only foods that have been thoroughly cooked and are still hot, or fruit that you have peeled yourself. Avoid undercooked or raw fish or shellfish. Make sure all vegetables are cooked. Avoid salads. Then avoid foods and beverages from street vendors. Some control methods for cholera are: the individuals affected with cholera need oral rehydration therapy with sodium chloride plus sucrose, then sodium bicarbonate and potassium chloride to stimulate water uptake by the intestine. And some antibiotics are also used like tetracycline or aprofloxacin. And the most reliable control method are proper sanitation, especially water supplies. Also, medical treatment can prevent dehydration and all other complications. The next important bacterial infection is Vibrio parahemolyticus gastroenteritis. Is a kind of foodborne illness caused by the bacterium Vibrio parahemolyticus. The main reservoir is seafood, and this bacteria can grow in the presence of 1 to 8% of sodium chloride, and the pH range is about 9.8 to 11, and the optimum range is about 7.6 to 8.6. They can cause symptoms from 3 to 76 hours after the ingestion of organism, and it is said that about 1 million organism consumption can cause to produce symptoms from mild to severe and fatal one. Intestinal abdominal pain, a kind of burning sensation of the stomach, then vomiting and diarrhea with watery stools, and sometimes cause bloody discharges and fever. And the list of foods involved in Vibrio parahemolyticus include: the vehicle foods for outbreak are raw, improperly cooked or cooked recontaminated seafood such as oyster, shrimps, crabs, lobsters, clams, and related shellfish. Cross contamination can lead to other foods as vehicles. Improper refrigeration of seafoods contaminated with this organism will allow its proliferation that can increase the possibility of infection. The bacterial infestation is diagnosed by culturing of organism from diarrheic stool of the individual, and it can be prevented by avoiding the consumption of raw or improperly cooked seafoods. Next, move on to the important bacterial infection called Shigellosis. This is a highly infectious bacterial disease caused by the Shigella species that mainly affect gastrointestinal tract. This Shigellosis is also known as bacterial dysentery, caused by facultatively anaerobic gram-negative bacteria, which is a non-spore-forming rod-shaped organism belonging to the genus Shigella and the family Enterobacteriaceae. This Shigellosis is a self-limiting disease, and the incubation period is about 5 to 6 days. It can last from 5 to 6 days if untreated. Young, malnourished children, elderly, and immunocompromised, that is AIDS patient, the disease may be fatal. And the mortality rate in case of Shigellosis is about 5 lakh children worldwide each year. They grow both aerobically and anaerobically, and also grow freely in warm, bland, and moist foods. This is a no flagella and non-motile organism. Example: Shigella sone, Shigella dysentery, Shigella flexneri, and Shigella boydii. About 10 colony-forming units of a Shigella dysentery are known to initiate infection individuals, and these organisms can tolerate salt concentration of 5 to 6% and are heat sensitive in nature. The contamination can be caused due to poor personal hygiene, then shellfish, fruits and vegetable, chicken, salad, and the mode of transmission is through oral fecal route. Let's discuss in detail about the symptoms of shigellosis. That is, they release lipopolysaccharide endotoxin which affect the internal mucosa that can range from mild to very severe and fatal. And the incubation period is about 1 to 7 days and maximum of 14 days. The main symptoms include abdominal pain and cramps caused by the inflammation of a mucous surface of large intestine, nausea, diarrhea, vomiting, and elevated temperature. The excessive diarrhea can lead to electrolyte imbalance that is due to the loss of water from the body and electrolytes can lead to ulceration in large intestine. In severe case, it can cause kidney failure, jaundice, and persistent internal bleeding. Shigellosis can be diagnosed by the Shigellosis can be diagnosed by identification of culture isolated from stool helps to diagnose the disease. Shigellosis can be diagnosed by the identification of culture isolated from stools. Some prevention and control methods for shigellosis are avoiding contamination of foods by animal or human carrier or their excretion. Then thorough cooking and prompt cooling is essential. Proper personal hygiene should be maintained. In severe cases of shigellosis, dehydration of body can cause intravenous replacement of fluid with electrolytes. Then ampicillin antibiotic can decrease the duration of the disease. Then another bacterial infection, Campylobacteriosis. This is a type of bacterial infection caused by Campylobacter species, typically Campylobacter jejuni. This is a gram-negative rod bacteria, the microaerophilic organism. That is, they require only low level of oxygen. These bacteria can be isolated from healthy cattle, chickens, birds, and even flies. Other sources include non-chlorinated water, streams, and ponds. This bacteria can cause between 5 to 14% of all diarrheal illness worldwide. This Campylobacter jejuni mainly affect children under the age of 5 years and young adult of about 15 to 29 years old. The major symptoms include diarrhea, watery or sticky, and contain blood and fecal leukocytes, that is white cells. It also causes fever, abdominal pain, nausea, headache, and muscle pain. And this disease can occur from 2 to 5 days after ingestion of contaminated food and water. This condition can last for 7 to 10 days, but relapse are not common, about 25% of the cases. Relapse meaning reoccurrence of the same symptoms of diseases. The infected dose of Campylobacter jejuni is considered to be 400 to 500 bacteria in number. And the major food associated with Campylobacter jejuni is contaminated raw. And a survey shows that about 20 to 100% of our retail chickens are contaminated. Then raw milk and non-chlorinated water is also one of the source of. These bacteria are often carried by healthy cattle and by flies on farm. The prevention method for Campylobacter jejuni is wash hands before preparing foods, wash hands immediately after handling raw poultry or other meat, then proper cooking of chicken to internal temperature of about 170 degrees Celsius, then drink pasteurized milk and chlorinated water. Then wash hands after handling pet feces or visiting zoos. Some other control measures include antibiotics such as ciprofloxacin or azithromycin. This erythromycin also helps to treat diarrhea. Also, having diarrhea should take plenty of water. Next one is yersiniosis, that is Yersinia enterocolitica infection. And Listeria enterocolitica, which is mainly contaminated through food. Yersinia enterocolitica is a bacterium that can cause a range of diseases, include gastrointestinal infection, septicemia, and reactive arthritis. The major characteristics of Yersinia enterocolitica are gram-negative bacteria, then facultative anaerobe that can grow with or without oxygen. Then they are motile with the characteristics of bulldozer motility pattern. Then psychotrophic, that is, it can grow at a refrigerated temperature and make the food unsafe. And the occurrence of yersiniosis is mainly in animals from Yersinia enterocolitica, which is isolated, include cat, birds, dogs, beavers, guinea pigs, rats, camels, horses, chickens, deer, cattle, swine, fish, and oyster. These are widely believed that swine constitute the single most common source of Yersinia enterocolitica in human. The organism can also be isolated from cakes, vacuum packed meat, seafood, vegetable, milk, and other food products, also from beef, lamb, and pork. And the swine appears to be the major source of pathogenic for human. Development of symptoms can take several days after ingestion of contaminated foods, and it is characterized by abdominal pain and diarrhea. The children appear to be more susceptible than adults, and it is said that this organism can be present in stool for up to 40 days following illness. Then severe abdominal pain, fever, and diarrhea occur from 24 to 36 hours after consumption of the infected product. The abdominal discomfort is quite as sharp pain in the lower right quadrant of abdomen. Next, foodborne infection. The next bacterial food infection is listeriosis, that is Listeria monocytogenes infection. This is a gram-positive, motile, facultative, anaerobe, and psychotrophic bacteria found on decaying vegetation and in soils, animal feces, sewage, silage, and water. In cattle, it can result in abortion and mastitis, and infected animals shed the organisms in milk. Some other infected organisms include sheep and chicken that can serve as a source of infection in food supply. This listeriosis in human is not characterized by a unique set of symptoms, as the course of disease depends upon the state of host. When susceptible adults contract the disease, meningitis and sepsis are the most common recognized symptoms. Pregnant females who contract the disease may not present any symptoms, but when they do, they are typically mild and influenza-like. In pregnant female, it can cause abortion, premature birth, or stillbirth. In newborn, it can cause meningitis and they begin from 1 to 4 weeks after birth. In case of adult, it takes about 1 to several weeks. So, thanks for watching the entire video till the end. For more classes, do like, share and subscribe to Factscribe. Thank you.